Hello, I am Dr. Y.S. Rajput, working as a emeritus scientist, Animal Biochemistry Division, National Dairy Research Institute, Karnal. Welcome to EPG Pathshala. Today, I am going to teach you about one of the important techniques in the field of molecular biology, that is recombinant DNA technology and its use in expression of genes. The technique is widely used in all disciplines of life sciences. This technique is increasingly applied for characterizing genes and making useful products economically. Many products for the medical use are in market, and these are prepared by employing recombinant DNA technology. Recombinant DNA technology can be defined as the technology that combines uses of different enzymes to cut and paste DNA sequences of interest and form a new recombinant DNA sequence. The recombinant DNA sequences thus formed can further be transported through specially designed vectors that carry DNA into host cell. The recombinant DNA present inside the host cell can be multiplied or translated to a specific protein using the host cell machinery. I hope you will derive benefit from this lecture for understanding recombinant DNA technique. In today's lecture, we will understand recombinant DNA technology and its application in expression of prokaryotic gene. First, I will explain in detail what are the tools of recombinant DNA technology. The other objective of the lecture is to explain basic steps involved in recombinant DNA technology. I will also be explaining the design and protocol for expression of gene. The present slide shows the concept map that will be followed in this lecture. The lecture is broadly divided into 3 sections. In first section, we will now know that what are the requirements of recombinant DNA technology. These could be enzymes, vectors, DNA inserts, hosts, and many other requirements. And in second section, we will focus on understanding the major steps involved in recombinant DNA technology. In third or last section, we will discuss the bacterial or prokaryotic gene expression system and its advantages. Genetic engineering is referred as the manipulation of genes isolated from an organism to alter some specific characteristics attributed by the gene. Genetic engineering finds tremendous applications in the field of medicine, agriculture, and industry. Recombinant DNA technology is a component of genetic engineering. It refers to the process of cutting a known or a target DNA sequence from one organism and introducing it further into another organism, thereby altering the genotype of the recipient organism. Recombinant DNA, or popularly referred as the rec-DNA, is a form of artificial DNA that is made through combination of one or more DNA strands or insertion of one portion of a strand into another, thereby leading to a combination of DNA sequences within the different species. In the present slide, some of the key elements required for recombinant DNA technology have been listed. These are enzymes. Different enzymes used for recombinant DNA technology are restriction endonucleases, exonucleases, DNA ligases, DNA polymerases. Other requirements are cloning vector, host organism, DNA insert or foreign DNA, linker and adapter sequences. Restriction endonucleases are essentially required for recombinant DNA technology. These enzymes cut DNA sequences at specific sites. The cuts or cleavage in DNA strands are made only within or near some specific sites called recognition sites or recognition sequences or restriction sites. Recognition sites are specific for every restriction enzyme. There are three main categories of restriction endonucleases, namely type 1, type 2 and type 3 restriction endonucleases. Different types of restriction enzymes differ in their properties. Type 1 restriction endonuclease cleaves only one strand of DNA. These enzymes have the recognition sequences of about 15 base pair length. Type 2 restriction endonucleases are most stable. These nucleases cleaves only at specific sites. These nucleases produce DNA fragments at a defined length. These enzymes cleaves both strands of DNA immediately outside the recognition sequences. Type 2 restriction endonucleases are most suitable restriction enzymes for gene cloning. Type 3 restriction endonucleases are not used for gene cloning. These are the intermediate enzymes between type 1 and type 2 restriction endonuclease. The cleavage pattern of different groups of restriction enzyme is different. DNA cleavage in type 1 restriction endonucleases takes place about 1000 base pair away from the 5' end of the sequence TCA, which is located within the recognition site. For example, ECO K or ECO B. The recognition sequences for type 2 restriction endonucleases are in the form of palindromic sequences with a rotational symmetry. A palindromic sequence is a sequence that is the same when read from 5' to 3' on one strand and 3' to 5' on other complementary strand. Examples of type 2 restriction endonucleases are HIND1, ECOR1, PU2, LV1, HE3. Type 3 restriction endonuclease cleaves the DNA at a well-defined sites in the immediate vicinity of the recognition sequence. Example is HINP3. Restriction enzymes require magnesium ions and ATP for their functioning. Type 1 and type 3 restriction endonucleases require both magnesium ions and ATP for their functioning, whereas type 2 restriction endonucleases require magnesium ion and do not require ATP for their functioning. Restriction endonucleases can cut the DNA molecule in two ways. They can cleave the two DNA strands at two different points. Such cuts are termed as staggered cuts, and this results into the generation of protruding ends or sticky ends. Such ends can pair readily with each other upon their being provided with suitable pairing conditions. Another feature of the restriction endonucleases producing such sticky ends is that the two or more such enzymes with different recognition sequences may generate the same sticky ends. Generation of sticky ends is shown in left side of diagram. Restriction endonucleases can cleave also both strands of DNA simply at the same point within the recognition sequence. Thus, DNA fragments with blunt ends are generated. Pu2, He3, Lu1 are the examples of restriction endonucleases producing the blunt ends. Generation of blunt ends is shown in right side of the diagram. Exonucleases removes or cuts nucleotides from the ends of a nucleic acid sequence. The enzyme removes nucleotide from 5' or 3' end of a DNA sequence. Unlike endonucleases, these never produces internal cuts within the DNA molecule. Examples of some of the exonucleases used in recombinant DNA technology are Exonuclease Val3-1, Eco Exonuclease 3, Lambda Exonuclease. Functioning of exonucleases and endonucleases is depicted in diagram. The function of ligase is to ligate or join two fragments of DNA. This is achieved by the synthesis of the phosphodiester bond. In recombinant DNA technology, DNA ligases are used for sealing nicks between adjacent molecules. DNA polymerases are associated with the synthesis of new complementary DNA strand of an existing DNA or RNA template. Examples of some DNA polymerases used commonly in genetic engineering are DNA polymerase 1 prepared from E. coli, Taq DNA polymerase used in polymerase chain reaction. Reverse transcriptase uses RNA as a template for synthesizing a new DNA strand called cDNA or complementary DNA. Its main use is in the formation of cDNA libraries. Other enzymes which are used in recombinant DNA methods are terminal deoxynucleotidal transferase. This enzyme adds single-stranded DNA sequence to 3' terminus of the DNA molecule. One or more deoxyribonucleotides, for example, deoxy-ATP, deoxy-GTP, deoxy-TTP or deoxy-CTP, are added to 1 by 1 to 3' end of the blunt end fragment. Alkaline phosphatase removes phosphate group from the 5' end of the DNA molecule. Polynucleotide kinase adds phosphate group to the 5' end of a DNA molecule. Cloning vector is a DNA molecule that has the capability to replicate within the host organism. The target DNA is introduced into this vector to produce recombinant DNA molecule. A great variety of cloning vectors are available for use with E. coli as the host organism. Sometimes it becomes desirable to use different hosts for cloning experiments. So, various cloning vectors have been developed based on other bacteria like Bacillus, Pseudomonas, Agrobacterium, and on different eukaryotic organisms like yeast and other fungi. Different types of DNA molecules may be used as a cloning vehicles such as plasmids, bacteriophages, cosmids, phosmids or artificial chromosomes. Plasmids are circular double-stranded DNA molecule found mostly inside E. coli. Plasmids are also commercially available. A host organism is important because it facilitates the replication of the recombinant DNA molecule. Most widely used host for recombinant DNA technology is E. coli. This is because cloning and isolation of DNA inserts is very easy in this host. A good host organism should be easy to transform and in which the replication of recombinant DNA is easier. The desired DNA segment which is to be cloned is called as DNA insert or foreign DNA or a target DNA. It can be viral, plant, animal or bacterial origin. Dear students, it should be clear that recombinant DNA technology deals with cutting of DNA and fragments so obtained are ligated to new DNA molecule. The enzyme which exhibits specificity in cleavage are useful in recombinant DNA technology. It will enable their ligation in a vector easier. A range of restriction enzymes are available for creating sticky or blunt ends, and this will enable to select strategy for ligation. Enzymes required for recombinant DNA technology are commercially available and there are proving very handy for researchers. Linkers and adapters are short DNA molecules and are added to cut ends of DNA for creating desired modification for ligation purpose. In next section, overall methodology of recombinant DNA technique will be explained. The other aspect which will also be explained is prokaryotic expression system. Linkers have one or more target sites within them for the action of one or more restriction enzymes. They can be ligated to blunt ends of the foreign DNA or vector DNA. Then ligated DNA is treated with a specific restriction endonuclease to produce cohesive ends of the DNA fragments. Cohesive ends in the DNA fragments are created to allow proper complementarity with the vector with its cohesive ends. EcoR1 linker is a common example of frequently used linkers. This concept is elaborated in diagram. Adapters are molecules which have been synthesized chemically and designed to possess one or both sticky ends. The foreign DNA is ligated with adapter on both ends. This new molecule so formed is then phosphorylated at the 5' termini and then integrated into vector DNA to form the recombinant DNA molecule. Figure shows the role of adapter sequences in recombinant DNA technology. There are 6 main steps in recombinant DNA technology. Step 1 is identification and isolation of gene or DNA fragment of interest which is to be cloned. This desired DNA fragment or segment is then isolated enzymatically by restriction endonucleases. This DNA segment of interest is termed as DNA insert or foreign DNA or target DNA or clone DNA. The target gene to be cloned can be obtained from various sources such as genomic library, cDNA library, chemical synthesis of gene, or using techniques for gene amplification such as PCR. Step 2 is selection of a suitable cloning vector. Most commonly used vectors are plasmids or bacteriophages. The size of the gene fragment which is to be cloned often determines the selection of vector. Step 3 is the joining of target gene into select vector to construct a recombinant DNA. The target DNA or DNA insert is ligated to vector DNA to form a recombinant DNA molecule by DNA ligase. The recombinant DNA molecule is often called as cloning vector insert DNA construct. Figure shows the use of DNA ligase to form recombinant DNA molecule. Step 4 is the introduction of recombinant DNA into a suitable host. The recombinant DNA molecule is introduced into suitable host cells. The selected hosts are bacterial cells like E. coli. Sometimes yeast and fungi may also be utilized. This process of entry of recombinant DNA into the host cell is called transformation. A recombinant vector can be introduced into host cell by different gene transfer methods. This deals about the different gene transfer methods. Physical gene transfer methods include electroporation, microinjection, gene transfer mediated by liposomes, gene transfer mediated by silicon carbide fiber, gene transfer mediated by ultrasound, and DNA transfer by pollen. Chemical gene transfer methods includes gene transfer mediated by polyethylene glycol, gene transfer mediated by calcium chloride, gene transfer mediated by DEAE dextran. Imbibition of DNA by cells, tissues or organs. Other gene transfer methods include gene transfer mediated by virus. Step 5 is the selection of transformed host cells with gene of interest from among the non-transformed cells. Various methods are employed for separation of the transformed cells, that is the host cells which has taken up the recombinant DNA molecule from the non-transformed ones. These include antibiotic resistance, use of visible characters, performing assays for biological activity, colony hybridization or blotting test. Step 6 is the expression and multiplication of target gene in host. The selected cells are then cultured in large scale. The inserted gene within the vector that has been transformed will therefore replicate within the host to synthesize multiple copies of the desired gene. The figure gives a schematic representation of various steps involved in recombinant DNA technology. For expression of desired gene, an expression vector is used. An expression vector is designed for the expression of the protein which is coded by the clone gene. It is basically a cloning vector with specific suitable expression signals to facilitate maximum gene expression. The following expression signals are introduced to get maximum gene expression: strong promoter, strong termination codon, transcription termination sequence, translation initiation sequence, adjusting the distance between promoter and the clone gene. When the target gene is cloned into expression vector and when expression system is the bacteria, bacterial expression of target protein occurs. This is also known as prokaryotic expression. The figure shows a typical prokaryotic expression vector with a clone gene of interest, an origin of replication, a promoter, and a ribobinding sites are positioned upstream of the target gene that has been cloned into this prokaryotic vector. Additionally, one or more transcriptional termination sequences are positioned downstream. The selection marker commonly used is a drug-resistant gene encoding an enzyme that inactivates a specific antibiotic and helps in selection of transformed cells over non-transformed ones. When the transformed bacteria are expressed to a specific antibiotic, only those that have been able to successfully take up the recombinant DNA or plasmid will survive. An example of the prokaryotic expression vector is PET3A vector. The host of this plasmid is BL strain of E. coli. The product is synthesized in mass culture in large quantities. This is how insulin is produced in large quantities in cell culture. In this lecture, you have looked at recombinant DNA technology and its use in prokaryotic gene expression system. In first section, we saw that enzymes, vectors, hosts, DNA inserts are required for recombinant DNA technology. In the second section, we have looked at the major steps in recombinant DNA technology. In last section, we have discussed the bacterial or prokaryotic gene expression and its advantages. Dear students, I must tell you that prokaryotic gene expression system has enormous advantages over eukaryotic gene expression system. My presentation has limited to prokaryotic gene expression system. This system allows for obtaining large quantities of recombinant proteins in a short time. Culturing of bacteria is simple and inexpensive. The simplicity of genetic modification and its availability of many bacterial mutants. I must thank you for paying attention to this one of the very important biochemical technique in the field of molecular biology, and I hope that you will enrich your knowledge for understanding the technique of recombinant DNA technology, and if you get a chance to use in your research profession, you will be able to help and get a good exposure to this technique, and will be able to make use of this particular technique in a better way. I must thank you for paying attention to this lecture.